Bismillahirrahmanirrahim. Hello, everyone. I hope everyone is fine. Today, I'm going to talk about another tool that is to be used for design of sgRNA. Previously, I have recorded several videos and I have shown live seminars for designing of sgRNA. Then several days ago, I met with another tool, and I think it's very important and it's a powerful tool. So I thought I could share with you. The basic question is, what is CRISPR? What is Cas9? What is Cas13, CPF? If you are interested in learning these basic things, you may please refer to my previous videos, or you can search some literature, read some papers from Jennifer Doudna, from David Leo, from Feng Zhang, and there are so many other researchers. They have published a lot of data. Simple answer for what is CRISPR is, this is a system that is modified for gene editing. And now the gene editing is so simple that even a bachelor or even a high school student can make a change in the genome of like human cells or mice cells or some insects like mammalians or some plants. Before going to the design CRISPR design tool, which is here, it's from Sigma Aldrich. I would like to show you these two PPTs, these two slides. Here we can see the purple, which is the genomic DNA. Genomic DNA means this can be the DNA from human cells, from plants, from animal, mammalian, whatever. But that is, in other words, that is our gene of interest. In the green, dark green and light green, you can see this is Cas9 enzyme. Cas9 enzyme is an endonuclease that makes DSP, double strand break. There is a very common question regarding the Cas9 that we already knew there are so many restriction enzymes, endonucleases, that cut the DNA. Then why we needed the Cas9? The explanation is very complex, but I have a very simple answer for it. And that answer is, for the other endonucleases, the other restriction enzymes, we cannot guide them. We cannot take them to our region of interest. But for the Cas9, we can control it. We can guide it with the help of a matching gene sequence that is called as guide RNA. Usually this is called as sgRNA because this is the synthetic form of trans-activating and trans-activating RNA, like track RNA, CRRNA and track RNA. This is synthetically combined, and then we can target this Cas9 to the region of our interest in the gene of our interest. How we can guide it? We can guide it by using a PAM, palindromic sequence. In simple words, for the case of Cas9, that PAM is NGG. Means N can be any nucleotide like ATCG and GG are the two guanines. The next slide shows you the map of a plasmid. In this plasmid, there are several things since this lecture is not based on the explanation of the plasmid, so I'm not going to explain the segments. I'm just wanna go to show you the Cas9 over here. This will make a double strand break. And another thing I'm more interested to show you is the gRNA scaffold. It means that you have to digest this plasmid with the restriction enzyme, which is present here, most likely BBS2, BBS1. And here you can see the U6 promoter, and then we have the scaffold for the guide RNA. That guide RNA, now I'm going to show you how you will design it, will take this plasmid to your gene of interest, and then inside the gene of interest, it will take you to the region of your interest. So you can make a knockout at a double strand break in your gene of interest. Usually, I will suggest you to use two plasmids for as two sgRNAs because that will make two breaks, and then you can delete the fragment. Now let's go towards the tool. If you're interested, you can read this paper also. This is like a very brief explanation, and it's not a new one also. You can just go to the Google Scholar, and you can just type the CRISPR-Cas9, and there is a lot of papers you can see, and the updated one like the base editing, like the prime editing, like the search and find and research and replace and enhance and so many things. As I have talked about the region, the PAM, this PAM is protospacer adjacent motif, NGG. What is the function of this PAM? It will control the guide RNA. And when this guide RNA is located by this PAM, this Cas9 will recognize this region. And there will a bubble will form in the gene of interest in the DNA simply, and this Cas9 will make two cut. This Cas9 has two restriction sites. One is NH8, and I just forgot the name of the another restriction site. So there is another version of this Cas9 that is called nickase, which will make a single cut, and the third form of the Cas9, that is dead Cas9, that is catalytically deactivated. Both the restriction and sites are deactivated, and that will not make a double strand break. That instead, that will just take the machinery to the site of interest, and in that case, we can do the over expression or the down regulation of the gene. Now let's go toward the CRISPR design tool. Here, you can just take this link from the description. I will provide it over there. Here you need to input your information, and based on this information, you will get your sgRNA. And that sgRNA, as I have shown you here, this plasmid, you will ligate that sgRNA over here, and that's it. Here I'm going to give you an example of a gene that is called BEX2, Brand Expressed X-linked 2. It asked me, how would you like to choose your genome of interest, Homo sapiens or the mus musculus? I'm always interested in the mice, so I choose the mus musculus, and then CRISPR design based on gene symbol, gene ID, or you wanna copy paste. You have the sequence and you wanna paste it here. I would like to go for the gene symbol, and then you have to tell them about the gene, that is it a protein coding gene, or it is a microRNA, or long non-coding RNA. This gene is a protein coding, so I will keep it as a default. Enter gene sample BEX2, and here it comes a lot of information, but I have to choose my gene of interest. If I cannot see my gene of interest, sometimes these genes have different name, then I will write the full explanation of the gene. Brain expressed X. It is not taking my gene, so I have to refresh the page. I refresh the page, select genome, musculus, then gene sample, type of gene is protein coding, and here I will type EEX2. You see that I cannot find that gene name over here, so there is uh, a more easy way. Let me use that way. So I will go to NCBI nucleotide, NCBI. Here I will type PX2 musculus. Yep, it comes. So my gene ID is 12069. I will copy and bring it here. Yes, the gene come over here. I will choose this gene and wait. Okay. My mRNA is 902 bases. It's a small gene. I have worked on this gene. I have the mouse model and I have published several papers. Cas9, which Cas9 you wanna go to choose? It's NGG. You want to make a double strand break, or it's a nickase. I want a NGG. And the guide RNA mismatches. What is mismatches? That means off targets. Number of mismatches are potential off target sites in the genome. And then submit. The beautiful thing of this CRISPR design tool is what? Let me tell you. The beautiful thing in this CRISPR design tool is it will also provide you the upstream and downstream primers, which is not available in bench-link. Here you can see your target and your sequence, then your PAM is TGG, your this sgRNA is present on sense or antisense, so it's on sense, chromosome X already known, start from here, and then transcript hits, your efficiency, your specificity, and then a number of sites, then it tell you the off targets. So if you click on this view, it will show you the possible off target sites mismatches. If you want to go to more details, you can click over here, and the mismatches are highlighted with the red font color. You can download this also. Okay, come over here. It shows you the primers. So this is very beautiful. Exclude off-target search. Yes, of course, I want to exclude, but you can include it also. If you want to do so, you can include. But here I would like to exclude off-target and submit. Then this will give me, oh, here are no primers available. Then you can ignore this sgRNA and you can go to another one and you can look for it. Now Ricard's form. You know why? Because this sequence, the sequence of this gene is very troublesome and it does not qualify the standard operating procedure SOPs for the design of the primers. So then in this case, you have to design your primers manually by sacrificing some rules from the standard operating procedure. Anyhow, this is a new tool to design your sgRNA, and here you can get more information about your genome. It will tell you everything, and you can get the gene details also, BX2 and description, whatever you want, and then you can go to NCBI or Ensemble or the other genome browsers, so you can get the more information. You play, you can play with this, all these tools: CRISPR design, off-target, batch design, donor design, publication data, and you can have a look at this website. I hope you have enjoyed the session for today. And if you have any question, please don't hesitate to contact with me. I have provided my WhatsApp number, my email. You can follow me on my Facebook. And if you have any question, you want to discuss your project or whatever, you can contact with me. That's all for today. Bye.